Okay, so welcome everyone, and thank you for joining us virtually for the latest version of the Variant Effect Seminar Series. My name is Jan. I'm a member of the VES Organizing Committee, along with Adriani de Souza, Diego Calderon, Ohana Bezerra, and Mariano Martin. These seminars are made possible with support and a lot of help from Lara Muffley, Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effect Alliance. As a reminder, we are always on the lookout for potential speakers for future seminars. So if there is anyone you would like to see present, or if you're interested in sharing some of your own work, then feel free to submit a speaker nomination form. As usual, before starting presentation, I have a few housekeeping items to mention. Each talk is around 20 to 25 minutes, but there should be time for questions. If you can feel free to post questions at any time in the Zoom Q&A, and we'll read them out loud to the speaker at the end. If you have additional questions after the end of the seminar and you are a NAVE member, feel free to visit our seminar series Slack channel for more discussions. And finally, we're posting updates on our Twitter account, so be sure to follow us there. You can catch the seminar today and previous presentation that have been recorded on our affiliate CMAP YouTube channel. So all the links will be provided for this. All the details of the seminars can be found on our website, including regularly updated schedule for upcoming speakers. And we have an announcement as part of the Alliance new protocol.io space. Interested members may sign up to receive a free protocol conversion code. The winners will be announced sometimes this summer. For the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. Our first speaker is Raquel Cuella Martin. She is an assistant professor in the Department of Human Genetics at McGill University. Her lab applies cutting-edge, large-scale precision genome editing to address outstanding mechanistic questions in the DNA damage response and their association with human disorders. Raquel undertook her PhD studies in the lab of Dr. Junior Chapman's lab at the University of Oxford. During this time, Raquel described the mechanistic role of the DNA repair precision protein 53BP1 in optimal p53 tumor suppressor responses. In her postdoctoral work as an EMBO long-term fellow at the Shisha lab at Columbia University, she used CRISPR-dependent base editing to perform genetic screen at nucleotide resolution and functionalize DNA variant at scale. Such an approach allowed her to identify loss, gain, and separation of function mutations and new functional domain in DNA damage responses protein, and to functionize variant of certain significant in cancer predisposition syndromes. The title of our presentation is Precision Genome Editing to Study Protein Function. Raquel, whenever you're ready.
All right. Okay. That's the one share. I hope that works. All right. Well, thank you for having me here. There were a couple of spoilers alerts already in that introduction, but hopefully it is still interesting. Thank you very much for that kind introduction. Thank you for the invitation. So yeah, my lab, I said, we use precision genome editing screens. At least we try. It's still early days, but we aim to become a screening lab. And today, I guess I'll go back as that intro was saying to.
Raquel, I think you have presenter mode. I think you're showing up presenter mode.
Okay. What is going on? All right. Thank you so much. I'm gonna try. Okay, maybe because I was sharing the desktop. Okay, share screen. Okay, share.
Yeah, it's still presenter mode, but
For whatever reason. Okay.
Go to the top right corner. There's like a flip screens button. That's worth trying.
yes, now
Okay.
it's fixed.
Yes, I was sharing. Okay, sorry about that, everybody. So I think I'm becoming one of those researchers that have been working on the same protein since they started their career, and then they just go on and on about it. So this is the protein that I worked on during my PhD, and that I continue working on at the moment, and it's called 53BP1. And for those of you that might be out of the DNA repair field, 53BP1 is a scaffolding protein, and this large protein. It has a very large unstructured N-terminal domain with an STQ rich that receives phosphorylation marks for the big DNA repair kinases. And then after that domain, you have like a couple of oligomerization motifs and two very key domains that are called the tandem tutor. And the tandem tutor recognizes methylation in H4K20. And remember that for later. And then it has a ubiquitin-dependent recruitment motif that recognizes H2A K15 ubiquitination that is a mark of DNA damage. And all these domains are important for 53BP1's role in non-homologous chain joining. And that happens physiologically in class switch recombination. We have some projects in the lab looking into that. But also this non-homologous chain joining is the pathway that drives the genomic instability in BRCA1 deficient tumors. And at the very C-terminal of this protein, it has a tandem BRCT, which is traditionally conceived as a phosphobinding module. And this was, everybody says, largely dispensable for the DNA repair activities. But however, it interacts with p53, which is the tumor suppressor for excellence, and then the deubiquitinating enzyme USP28. And that was what I worked on during my PhD, on how these three proteins come together to prime p53 for optimal tumor suppression. And that is important in the context of tumor suppression, in the context of the checkpoint response, but also when you have extended mitotic divisions and, caused by central duplication failure, you want to elicit cell cycle arrest to deal with that. And why I keep insisting on studying this is because when I get asked by my colleagues, it's like, okay, is your pathway mutated in cancer? And I'm like, damn it. So 53BP1 is not mutated in tumors. And we cannot really put a finger on how these two proteins are regulating p53, and if it's important, you will anticipate that they will have a tumor suppressor role. But then when we go to the DepMap database and we look at how these two proteins, 53BP1 and USP28, behave compared to p53, we see that they are the top correlators phenotypically, like right there. So you can see there in the bottom panel that you have, when you knock out p53, you get survival advantage, and you get a very similar pattern when you knock out 53BP1 or USP28, albeit to lesser magnitude. And this correlation is really nice, and also correlates well with known positive regulators of p53. So we're like, okay, this is, in the field, nobody will argue with ATM and CHEK2 being positive regulators of p53. So why this conception of this pathway being important is just not there yet. And recently, a group gave us a helping hand on those discussions when they found that USP28 is also deleted in tumors. And this deletion in tumors that have p53 wild type still phenocopies a p53 loss, similarly to ATM deletion or to the amplification of negative regulators such as MDM4. So we were like, okay, we think this is important for tumor suppression. We think this is a top positive regulatory mechanism. So we are going to keep insisting. And plus is my favorite protein. So I was like, we need to keep insisting here. So the way I was studying this protein back when I was doing my PhD was, we'll think like a very traditional manner, right? So we were browsing for literature evidence. 53BP1 was identified as a p53 binding protein. So we were like, there must be something there. And we found a screen and a few years ago, like already, I don't know, almost 20 years ago, that identified 53BP1 as a mediator of NATLin3 toxicity. And being NATLin3 a p53 stabilizer, you see in the ranking there, p53 as the top hit. We thought, okay, this is functionally relevant. And also in the top hits, but lower, was also USP28. So like, okay, these things are coming together. There are observations scattered in the literature that actually are pointing to this being important. So back when I started my PhD, which I'm old already, was 2014, with the CRISPR was like taking off, and we made some cell lines using CRISPR-Cas9, knocking out p53 and 53p1. And we saw that indeed, the magnitude of the effect was not that of p53 loss. I feel like if that was the case, we would have discovered it earlier, but it was an intermediate regulation. So p53 was really not able to transcriptionally activate or repress their target genes when 53p1 was missing. And then we went on to doing some like this very, this like little structure function analysis of making mutations in the domains that you know, and looking at phenotypes and also biochemistry. And this is when we came together, like, okay, it's the BRCT that is interacting with this complex, and that interaction is absolutely required for p53 to work. And on the basis of that, generated mouse models that separated the functions of 53BP1 in DNA repair and in p53 dependent responses, and we're able to recapitulate some of the phenotypes that we were seeing in vitro, which was very encouraging. But then, when you work in like this molecular biology and you just want to know how things work. And my PhD went fairly well, but then I was always just hitting my head against the wall with like, how does this work? How 53p1 and USP28 regulate p53? And I was encountering some challenges to do this little structure function analysis, because the proteins were not very easy to express ectopically for various reasons. They were large for structure function analysis, and they have these multi-domain architectures. So I was like, okay, if I express USP28 and try to do a deep mutational scanning, that was giving me all sorts of artifacts, like USP28 tends to overexpress, and that doesn't recapitulate. So I was like, okay, didn't really have the tools to inquire this. And then I moved to my postdoc lab, I had to give up on that. And then I moved to the postdoc, and which was 2018. And that was when, the base editing field was starting to just go all in. And we were like, okay, conceptually, the base editing should work on a screening format. So we set out to do that and generated some cell lines that were expressing B3 back in the day. I think now we are in B whatever max, but back in the day this was the best choice, is the FNLS B3. And to make those C to T transitions in defined windows of activity marked by the guide RNA sequenced, and with that being able to make 32 amino acid substitutions and stop codons. Right, so we made this in actually the mammary epithelial cell MCF10A, and then we had a lentiviral tiling library, do every possible NGG PAM, but that lentiviral tiling library targeting many DNA repair genes, including 53BP1. So I was like, okay, let me drop it there. This has been a tricky one to study, so I'll do that. Then we put the cells on the presence of DNA damaging agents, which obviously will uncover some phenotypes since we are dealing with DNA repair proteins, and then plotted our data, with more high throughput to look at how the different guys were behaving against different treatments, but also on a gene by gene basis to look at where those mutations localizing. Okay, so if we look closely at the results of that screening 53BP1, we can see that the nonsense and splice variants were giving us a survival advantage. And back in the day, I was like, well, this is not consistent with losing a repair function, but much more consistent with losing checkpoint activity, with having a p53 dysfunctionality. But what came out very interesting from that screen is like, if you have a survival advantage with a loss of function, then you will anticipate that things that give you a survival disadvantage could be gain of function mutations. And this is always very interesting because at least in my field, we don't find them very often. So I was like, okay, I was very interested by then. And if you had read the paper that is already 3 years old, I can't believe that. You can see that these, we focus on the tandem Tudor domain for several reasons, because this was a proof of concept study, and also because from previous work, I had the impression that this was involved also in the p53 regulation. And we picked some mutations. We picked a mutation that was targeting a glycine 1593 that looked like a gain of function behavior. And then we picked two mutations on the other side, one targeting G1560 that was targeted by 5 different guide RNAs, so really showing up, and then valin 1544 that was targeted by 2 different guide RNAs. And the initial validation that we do from one of those screens is a 2-color competitive growth assay. And basically we mix cells that are expressing a neutral guide RNA targeting AAVS1 with color with them cherry, and then we express a GFP tagged or express GFP in cells that express our guide RNA of interest. And then we mix them one-on-one and look at what overpopulates the other. So in this one-to-one assay, we saw that the things that gave a survival advantage in the screen were also given a survival advantage here. And our potential gain of function mutation, survival disadvantage in the screen, also a survival disadvantage here. And the mutations at endogenous locus were really made a high efficiency despite this being a diploid cell line. And so we trimmed out, teased out that the mutations that were giving drug resistance was valin to an isoleucine and a glycine to a lysins. And that strange mutation that was giving a gain of function sensitivity kind of thing was G1593K. And we went on and picked the structure of the tandem Tudor domain bound to H4K20 dimethyl, that if you remember from before, this is one of the interactions that brings 53BP1 to sites of DNA damage. And when we mapped our mutations, we color-coded by conservation analysis in blue variable towards pinkish conserve. And we put the mutations there, and they did not quite follow, all right, more conserve, like being, more susceptible to mutations. No, it was just a bit of random. But they were in general, like the 2 mutations that we are having a loss of function were at the rear interface, not really close to the pocket that was binding to the H4K20 dimethyl. And the glycine was very interestingly embedded glycine. So when we do targeted analysis and targeted structure function, we will never choose to mutate that. We will think that everything will get disrupted. And actually that was what the prediction softwares were telling us, that will cause a complete domain destructuring. Well, so then we went on to check what happened to p53 dependent responses when we had those mutations. And we used this, the very nice drug that we really liked, which is natlin3, to activate p53. And basically mutations that were giving us loss of function in the screen were survival advantage. We're also giving us loss of function here, survival advantage, just like a stop codon that we made. And then the one that was giving us a survival disadvantage, putative gain of function, was having the same behavior here. And, that's one of the things that when it works, we are very happy. The loss of function were actually losing the binding with USP28. P53 was bound just fine, but then the third partner in crime, that is USP28, was just lost there. And the one that was giving a gain of function, we're actually forming a complex that seemed to bind better. And we were having, better phenotype, better checkpoint, better complex formation, which made sense with that gain of function behavior. But then 53BP1, of course, has 2 functions. So we wanted to check whether those mutations could potentially be separation of function. And we went on to check in first whether 53BP1 was able to be recruited to sites of DNA damage. And it was just like a wild type control. So really that interaction with H4K20 dimethyl was preserved. And then we wanted to check in, is recruited, but is it functional? And the way we measure that is by looking at the restoration of homologous recombination in BRCA1 deficient cells. And you see that when you have a stop codon in 53BP1, you get RAD51 foci formation. But then when you have a wild type, that doesn't exist. Those cells are not able to assemble RAD51. And the same happened with our mutations. They were behaving like wild type. So indicating 53BP1 is going to sites of DNA damage and being active despite having these mutations that disrupt the other functions. So that was very interesting. And this actually was telling us, all right, we knew already that the BRCT was important. Now we know that the tutor is important. And how is that relevant? Well, because when you deal with proteins like these ones that are very large and not very structured, well, sometimes we find it tricky to understand how those interactions work. And this is how the AlphaFold 53BP1 looks like if you go to the AlphaFold database. So all that N-terminal and structure and all those loops are like just all over the place. It looks like a blob. But when we actually told like, okay, these are the two domains that are important, can we actually assemble a complex? AlphaFold did a much better job there. So we managed to assemble the tandem BRCT with the tandem tutor with P53 DNA binding domain. That interaction is known. But what was not known, it was how it interacted with USP28. So we managed to assemble the USP28 here. And good points about this is that the mutations that we were seeing in our base editing screen, we thought were an interface with USP28, and actually they are not. And with all our predictions, AlphaFold 2, AlphaFold 3, multi-unit complex, single unit complex, that interface is in the middle of an internal binding, or maybe 2 units of 53BP1 binding. It's in the middle of the tandem BRCT and tandem tutor. So we think that now when you disrupt that interaction, it's just basically disassembling the complex, but it's not a direct interaction. But what was really interesting from this structure was that we had the tandem tutor domain of 53BP1 interacting with a loop in the very extreme C-terminal of USP28. And that was interesting for what you will see in the next slide. But we also had the catalytic domain of USP28 binding the BRCT on the other side in an area that is traditionally the phosphobinding pocket. But we were looking at this and being a bit puzzled. Like some things made sense with what we had done before, but it looked like clearly that we were having a BRCT domain that is a phospholigand binding, having 2 phospho-independent interactions, one with P53 already known, and another one with USP28. And this one matched very well what we had seen before, because you have that phosphobinding pocket, 2 key residues equally important, and when we do the co-IPs, we see that we lose the binding with one, but not when we mutate the other. So that doesn't sit well with the phosphobinding interactions, and actually the AlphaFold was telling us that seemed to be a chart change interaction, not like a phosphodependent. But also something that I don't think has ever been reported is that we also have a methyl-independent interaction with the tutor domain. And in this case, it's with that loop over there. And for the binding of a methyl ligand, you need those 2 residues. And when we did our analysis, both of NATLI3 resistant for functionality and then the co-IPs, we see that the D1521 is needed, but not the W1495. So it looks like we have a complex that is using 2 modules, a phosphodependent binding-dependent module and a methyl-dependent module on like non-canonical ways. And we thought that was super interesting and very hard to find, which I think that might be one of my next slides, very hard to find with other traditional approaches that are a bit more guided. And then something that we can still not make sense of, and I'm sharing it with you here today, is that we had that in the tandem tutor and that we knew fairly well. But then we also had this other region that was right in the middle of the oligomerization domain and the GAR motif. And this region here, nobody has described any functional area, like any functional interaction or anything on that region. So we were a bit puzzled. But we validated the mutations that the screen was giving us and the guide RNAs, and everything held together: growth advantage, potential gain of function, growth disadvantage. And then we use the mutation at the endogenous locus so that we can inform our later co-immunoprecipitations and other experiments. And everything was inserted, fairly high efficiency. And we had a silent mutation that is very strange, and we still don't know what it does, but it was the strongest phenotype of all of them, particularly of our gain of functions. So we validated that. And then we did our classical approach of putting them on the structure, try to predict if there is a secondary structure and look at different sequence conservation. So in this case, AlphaFold tells us that this is a loop. And when we folded it in a different way with more flanking sequence, the initial part forms beta shed, next to the oligomerization domain, but other than that, no major secondary structure. And when we look at how the mutations are positioned, the loss of function are usually in conserved regions, but not quite fully, and the gain of function are just all over the place. So we cannot really make sense or draw a trend on that again. However, what we have very clear is that mutating those residues affects p53 dependent responses, loss of function from the screen on DNA damaging agents, loss of function when you put them in NADLIN3, and the gain of function, also gain of function when you put them in NADLIN3. But at the same time, they don't affect 53BP1 oligomerization, so it oligomerizes correctly and it gets recruited to sites of DNA damage correctly in the presence of those mutations. So we are still working here. We are yet to discover what this area of 53BP1 does. And I think what is interesting, and it was as I said, a spoiler alert on the introduction, these screens enable the systematic discovery of separation of function, loss of function, gain of function, and novel functional regions that was hard. They are hard to pick with traditional approaches or with sometimes the way we do the predictions. So now the way we deal with these questions, or the way we want to deal with these questions in the lab, are like using high throughput, either in silico or the screens that we run ourselves, to identify other partners in the axis, or what is the E3 ubiquitin ligase, because we are talking about the deubiquitinating event. Run more base editing screens to go identify all the interfaces that play a role in this complex, and also complement that with like thorough mechanistic analysis and also structural analysis to answer all those questions, like really how the complex is working, which still we don't know, but we will hopefully. And I think with that, I'm done. I think I'm on time or close. Which other questions are we interested in? Well, we just, the labs just are starting, so we are interested in a bunch of things. Very interested on trying to do this mutagenesis by mapping of allosteric sites and to identify novel protein domains, particularly in DNA repair proteins. And as I said, a bit more in the speculative end, trying to see whether we can do allosteric site targeting. And in another different area of the lab, I'm also very interested on the disorders that are associated with DNA repair and other rare disorders. So we are looking at possibilities for synthetic viable screens to identify targets for certain rare disorders, and we are doing some bits here and there on mutation correction for gene therapeutics. And since I moved to McGill, I'm trying to get everybody to run base editing screens. So I have gotten some collaborators interested, and we are doing some screens on different areas, for instance, to understand regulation of mRNA transduction, and translation, and then systematic analysis of uncharacterized open reading frames or poorly characterized open reading frames, and also some things that aren't already yet, but perhaps will have been a better fit for this seminar on characterizing variants of short-term significance in cancer predisposition syndromes. And with that, thank you to the lab and to all these young ladies that dare to share with me this adventure, and then our collaborators for bioinformatics analysis in the Talion lab, and then all my PhD postdoc lab and all other collaborators, and of course the funding agencies and you for your attention. Thank you. Thank you,
Raquel, for your presentation. Thank you so much. So if everybody has questions, please put it in the chat. We can start with a question that I have. I was curious. You told us that you have been working with this 53 BP1 for a long time in your career, because this is one of the top regions that are regulating like tumor, like cancer. And I was curious if this like the mechanisms of the DNA repair involved with this gene is also relevant for other human disease as well.
Okay, so the DNA repair function, well, I don't think that has been observed in patients, but mice that have 53BP1 deficiency have immunological problems because they are not able to perform class switch recombination or VDJ recombination. I don't think, up to the best of my knowledge and what I know of the field, if we have found patients with homozygous loss of function mutations on 53BP1 that I think will have an immunological deficiency. But what is important is that, it brings me to the other function of that, is that when, for instance, when you have people with BRCA1 deficient tumors that are treated with PARP inhibitors, that is a very nicely targeted therapy based on synthetic lethality, after a certain time you get the relapse with resistance, and usually it's because they lose 53BP1. So
Okay.
that is, I think one of the fields where you need to, when you look at the non-homologous enjoining pathway really closely, because it has some therapeutics implications, and in treatment resistance, or perhaps in treatment eligibility for those patients.
Okay, nice. And did you have the chance to look for the other like top genes from the figure that you showed, I think, in the third slide? You have other genes there, like ranking of the genes?
No, so in that field, in that screen, in the initial paper that I think you're asking me about the paper that looked at NALI3 resistance, the shRNA screen, or is it? No. So we are looking at ATM and CHEK2 as part of another project, but the role on p53 responses for that is very well defined. We are looking at whether we can identify separation of function mutations, as well as we did with 53BP1, which could be very interesting since they are cancer predisposing genes. But not much. There has been a recent paper actually looking at some other top genes, top correlators that we don't have there because they were a bit lower down, and that was very interesting. But in general, yeah, they were stability regulating the stability of USP28, so that's why they had phenotype. But no, we are just looking at ATM CHEK2 in a different context, so nothing much.
Okay, that's nice that you are deciphering a very interesting, important region in that sense. So I don't see any questions by now. Yeah, I see one from the Max Staller. When you design a screen, do you recommend targeting the full protein or targeting individual domains? Is the hit rate in the disordered regions lower than in the folded domains?
That's a very good question. Well, for 53BP1, yes. We always tile fully, unless we know that it's a specific region that we are interested in, that we already know that is involved in an interaction. When we are screening like this for discovery, we tile fully. We get more hits in functional domains, because also structural mutations that affect the structure tend to give phenotypes. But I will say that in my experience, we have found pretty interesting things in the disorder region. So I'm not willing to get them out of the screens. I will say, if somebody will ask me for the recommendation, I will say target everything unless you are, you know that your interest is X domain, and then I was like, okay, well, then you go for it. But yeah, and there is a lot in the disorder regions, perhaps, that we disregard with other approaches. So I like that.
Okay, great. I have one last question. It's about what you call separation of function mutations. I'm not very familiar with this term. It's not very common in the literature that I read, so we can explain a bit of it.
Well, separation of function. So in the DNA repair field, we have quite a bunch of these proteins that do different functions simultaneously, that have multi-domains and then do several things. So 53BP1 has a function on repair and a function on p53 regulation. So to be able to study those functions, ideally you will have a mutation that disrupts one but not the other, and one that disrupts the other one but not the first one. And those are what we call separation of function mutations.
Okay.
So the ones that I show here are affecting only p53 but not touching repair. So that allows us just to narrow down that function and understand their contribution and understand why that's important. Because if not, it's the same with a full knockout. You will have a confounding phenotype, just the sum of the loss of the two functions. So that's why we do this separation of function.
Got it. Thank you so much. Thank you so much, Raquel. In the interest of time, now we are going to pass the presentation for our next speaker.